Right, good morning guys. Welcome to another Biology with Miss Cherry. You'll be pleased to know we're moving away from the genetics, the calculations kinds of things, and now we're going to start looking a little bit more at populations. So to begin with, what can you see as a difference between those two dogs? They're the same species, so all dogs are part of the same family, but what causes those differences? And are the differences genetic or environmental? So pause and play again when you're ready. So as you can see, we're looking at variations in phenotype today. You could have guessed that from the starter. So we're going to be looking at why there are differences between individuals of the same species and how that can also be caused by environmental factors. As you can see at the top, it links very heavily to the end of module 4. So mutations, meiosis and genetic variations. And hopefully this will be a nice quick lesson. As you can see, it's only 16 slides long. Little bit of me doing stuff, little bit of you doing stuff, but jobs are good and should be over and done within about maybe half an hour all in. So we're going to start by taking a look at variation. So if you can think back to GCSE maybe last year, what is variation? So you're going to define it for me, do your best A-level definition, and then we'll check in a second to see how correct you are. So variation is the differences in characteristics between organisms, so looking at you and me and you and your teachers, due to genetic and environmental changes. And there are two different types of variation that we can look at. There's interspecific and intraspecific as well. Okay, so think about what you think those are. Think about what inter and intra means. Maybe you want to think about intermolecular and intramolecular. What do those prefixes mean in both of those cases? So pause and play again when you're ready to move on. So looking at these keywords, we've got a couple of different words that you might not have seen before, but you also might have. So interspecific variation is the variation that occurs between a species. And sorry if you can hear what sounds like a motorbike, it's the cat rubbing his face against the iPad. So how does a dog look different to a mouse, for example? How do we look different to chimpanzees? How do we look different to elephants? So between different species. Intraspecific variation, however, is variation within a species. So the differences between us as individuals in the human species, I guess. But we need to think how variations arise within species. So how does intraspecific variation arise? What is it that causes it? How can it change? How can it develop? And ultimately, you should probably recognise that we're moving towards discussing evolution. So have a go at summarising the notes on page 187. If you haven't got the book, I've taken a photo and uploaded it to OneNote for you on how genetic variation arises within a population. So you guys are going to feed back to me in your notes. I'm going to be able to see them. You're looking at random fertilisation, you're looking at meiosis and mutations. So you need to be as concise as possible. Cut the rubbish out. Okay, details, not waffle. And I would suggest that details, not waffle, is a principle that you take through life with you, because if you cut all the crap out, people are going to much prefer you in the way that you explain things to them. Now, before we move on, I want you to pause and think about this question and write down what you think is going to happen. Is there likely to be more or less variation in organisms, such as bacteria, who reproduce asexually? So pause, spend a couple of minutes thinking about this, discuss your answers in your notes, explain why you think this is, so if there's more or less, why, and then play again when you're ready to go through the answers. There's going to be less variation. And this is because that 2 out of 3 of the processes that are involved in inducing variation in a population, meiosis and random fertilisation, for example, are only applicable to sexual reproduction. So if we recap those terms, we know that meiosis is the production of gametes, and those are required for sexual fertilisation, so sperm and an egg coming together. And random fertilisation is the random joining of those gametes. If we're cloning ourselves, i.e. reproducing asexually, we're not going to get any of those gametes and the resulting fusion being produced. In asexual reproduction, only mutations are able to induce variation within that population. And we know that they're quite rare, but obviously within a bacterial population, they're going to reproduce quite quickly, and therefore there might be more incidents of these mutations occurring compared to in normal human cells. Now, while we move on, can you think of anything that is entirely genetic, that is a wholly genetic trait? So an example of a wholly genetic trait is blood group, and we've spoken about blood group before when we were looking at polygenic genes, and we were looking at how some of them can be co-dominant. So for example, type AB has IA and IB immunoglobulins on the outside of their cells. Now for the next few lessons, we've got to look at how we would graph or show these kind of data, these kind of characteristics on graphs and in various ways. So genetically derived traits are generally an example of discrete data, and that is to say that they fit into individual distinct groups. So you could be type A, type B. But what type of graph do you think you would use to show this trait? And what statistical test could you do to see whether the observed and expected ratios of each of those blood groups was statistically significant? So pause, have a think, maybe do some research and play again when you're ready. So for these kind of data, the discrete data, distinct groups of phenotypes or something like that, you're going to use a bar chart or a pie graph or a pie chart. And this is because it's to do with like proportionality in a cohort. Okay, and we're going to be able to use the chi-square test because the data is categoric. Remember, we could use the chi-square test when we're looking at phenotypes. Same we can do if we're looking at things like blood group. Now let's have a look at environmental variation, and we know that all organisms are going to be affected in some way by their environment. So the weather outside, maybe the food that's available, maybe the conditions that an organism lives in. But why are plants affected to a greater degree by their environment than animals? A fairly straightforward question. Give you about 5 seconds to think, and then I'll drag away the answer. Standard answer, plants are less mobile. We spoke about this when we were doing photosynthesis, when we were looking at taxes and kinesis as well. And it's because the plants are less mobile, they're not going to be able to get up and move away from their soil if it's a little bit dry, or they can't move to another area if there's more nutrients there, for example. So let's think about it in the context of a question that starts to link in modules together. So if I've got two rose bushes, both from the same parent plant, but I put one under my gazebo and I leave one out in the bright sun, which one will grow better and why? And with the why, I need you to think scientifically. Think back to module 5, think to what you've done. Why is one of them going to grow better than the other? So pause and play again when you're ready. So hopefully you will have recognised that the one that's in the sun, or the plant that's in the sun, is growing to grow better. And that is because there's more light. So we've got greater photosynthesis, specifically the light-dependent reaction, specifically the photolysis of that water, which is then going to help give us all of the electrons and stuff for the electron transport chain. And that's going to result in a greater rate of growth. Now, can you think of anything that is entirely environmental? So we spoke about things that are entirely genetic, being things like blood type. But what about something that is exclusively an environmental trait? There are lots of options, but the major one that I think of when I think of this is scars. Okay, scars are entirely caused by something that's happened around you or to you that leaves a physical mark. You might have also said maybe like accent or language spoken, but they're a little bit more variable because you can sort of teach them to yourselves almost, whereas scars are there, they're permanent. Now let's have a think about what happens when these two different types of characteristics start to interact. So imagine my parents are quite tall. Hey, I'm 5'3" and a bit. So if your parents are tall, You've probably got the genes for being tall. I've probably got those tall genes, but I'm not tall. Why is that? You could think about you. If your parents are tall, you've probably got the genes for tallness, but why won't you necessarily be tall? It's not that you won't, you might be, but why is it not imperative? And you can pause and play again when you're ready, or you can just carry on listening for the answer. And hopefully at the top of the slide sort of drove your thoughts in the direction that they should be going in. Things like height are combination factors. That means that they can be affected by genetics and environment. So if I've got the genes for tallness, that's fine, that's all well and good, but it can be affected by other things, like we've got down here, disease, maybe conditions that are affecting bone development, maybe you've got dwarfism, maybe you've got something like that, but also malnutrition can have an impact. So if you're not provided with enough nutrients to lay down strong bones, maybe you're not provided with enough nutrients to help your whole body develop, those are going to affect your height. Alternatives or other examples include things like skin colour. So your skin is the colour that you are born with, so maybe it's quite pale, maybe it's quite dark, but over time, environmental factors, things like UV, things like if you go and have a little suntan, things like if you enjoy a bit of fake tan and decide to, I don't know, turn yourself into a little what's-it, they are going to affect your phenotype as well. Hair, same example, you're born with a specific hair colour, hair texture, but you can change that. So there's lots and lots of incidences where phenotypic traits are not exclusively genetic or exclusively environmental, and that's something really important to bear in mind. So now, use the little photos of page 187 to 188 to fill out this table for me. You can pause and play again when you're ready. The next slide just has the answers on. So here we are with our answers. We've got an example for each of them all the way down, so it's all nice and filled out for you. Check your answers compared to what's on the slide. So genetic, an example was blood type. We went through that a few slides ago. And these traits, these genetic traits, generally have one gene controlling them. They are monogenic. Now, because things like blood type and these monogenic traits are grouped data, we can say that they are discrete, and we know that we're going to be using a bar chart or a pie chart or a pie graph or whatever you want to call it for representing them. Okay, conversely, for environmental, gone for height, which is a little bit of an iffy one, I know, because, you know, there's lots of different factors involved in them. And therefore, we say that it's a polygenic trait. There's more than one gene controlling them. Data-wise, environmental or environmentally influenced traits, we generally say are continuous, i.e. the data can be on a scale. Okay, so it can be in a range between A and B or whatever. And we represent these continuous data sets using a scatter graph. We do a scatter graph and a line of best fit, absolutely fine. And the data for these polygenic, environmentally influenced traits normally show a normal distribution. Now, I can't remember for the life of me whether this has come up before now. It might have done in module 4. But either way, we're going to recap it in the next few lessons, and we're going to look at normal distribution, what it means, and how we can represent these traits as a normally distributed graph. So the final thing we're going to look at before we do a little bit of a summary question is some background into scientific research a bit. So I want you to think about, why is it useful to study variation with identical twins? I want you to think about, what are identical twins and how are they formed? How are they developed? How are they made? So pause, maybe do some research, do some thinking, and play again when you're ready. So identical twins are generally used because genetically they're the same, and therefore any variation between them is likely to be environmental. So it's likely to be caused by differences in the way they were fed or brought up, or the differences in the activities they undertook. And this is because identical twins are formed by the splitting of a single fertilised egg. So in the early stages of fetal development, that egg is going to try and divide into two cells, and sometimes those two cells can break off from each other, and sort of embed themselves into different areas of the uterine wall, and that's how we get these identical twins. But because of that, they form two discrete individuals that are genetically the same. So now you guys are going to do a little bit of research. It says homework. As I said previously, everything's homework now. I want you to do some research about the study of the Jim twins. Okay, so it's called the Minnesota study of twins reared apart. That's what you're looking for. And I want you to answer these questions all the way down. So what was the study about? What did they find? Similarities and differences between the twins. What was so surprising? And why do the differences between identical twins increase as they age? And this last question is a link to Module 8. So you need to think about what you've been doing recently in your other lessons, or if you haven't covered it, maybe do some research to find that out. I'm not going to give you the answers for this. You guys are going to do it independently, and then I want you to be able to tell me in your notes about this study. So you can pause now and then play again when you're ready to go to the final slide. So the final thing we're going to do is some summary questions. As you can see, we're not doing any exam questions today because this short lesson links very heavily into other topics, and they've got a bigger set of exam questions to link together, so we'll just wait until those. But have a go at the summary questions on page 188 and then explain some of the causes of variation in human hair. So pause, and then when you play again, I'm just going to move the answer box away. So here are your answers, check them, and then we are done, guys. This has been a really short lesson. Thank you so much for watching, listening, taking part, doing whatever. I hope you found it useful, and I'll see you again next time. Bye!